Hello and welcome back to Rubisco for NEET. Today we will look upon semi-conservative DNA replication. So what do you actually mean by this term semi-conservative? Semi-conservative means partially conservative. So this scheme of semi-conservative DNA replication, it suggested that the two strands would separate and then each strand would act as a template for the synthesis of new complementary strands. So after this completion of the replication, so the DNA molecule would have one parental strand and one newly synthesized strand. So here as you can see, this is the parental DNA strand. So from here, one of the strand is coming and from here the other strand is coming. So from this one of the parental DNA strand, a new strand is being polymerized in 5' to 3' direction. And from the other parental DNA strand, you can see that another new strand is being synthesized in the same 5' to 3' direction. So therefore, what are this? These are the progeny DNA molecule. So this progeny DNA molecule has one parental strand and another newly synthesized strand. So this is why this is known as semi-conservative DNA replication, or there is a partial conservation of the parental character as well as new characters are being added. So now, who actually performed the experiment to prove that DNA replicates semi-conservatively? So it was Matthew Michelson and Franklin Stahl who performed the experiment, and you have to remember the year for NEET, that is 1958. So what actually they did? First of all, so we all know E. coli, right? Escherichia coli. So they grew Escherichia coli in a medium containing 15 NH4Cl. Now, as you already know that 15N is the heavy isotope of nitrogen and 14N is the normal isotope of nitrogen. And they used it for the growing of E. coli for many generations. That means the DNA strands, as you all know, this DNA strands contain the nitrogenous base, right? So that means this 15N was present in those nitrogenous bases. Therefore, 15N was incorporated into the strands of DNA which were being synthesized, as well as the other compounds which contained nitrogen. Now, but this heavy DNA molecule which was synthesized containing this 15N nitrogen, this could be distinguished from the normal DNA. How? By centrifugation in a cesium chloride density gradient. Now, what is this density gradient of cesium chloride? There will be a picture. I'll be showing it to you later on. Now, after growing E. coli in 15 NH4Cl medium, then they transferred the cells into a medium with normal 14 NH4Cl, that is the normal isotope. And then they took the samples at various time intervals of 20 minutes. And now the cells multiplied, but the DNA that they extracted, they remained as their double-stranded helices. Now they also separated independently on their cesium chloride density gradients to measure their densities of DNA. Now what it actually was, let's show in a picture. So now this is the picture I was talking about. So this was the first step. They incorporated the E. coli cells in 15 NH4Cl. That means this heavy isotope. So this heavy isotope nitrogen was incorporated into the DNA because, as you know, DNA contains nitrogenous bases as well as various other substances with nitrogen. Now, after that, they also introduced the cells in 14NH4Cl, that is the normal ones. So now those nitrogen were incorporated into the DNA. So what actually happened after 20 minutes? Now this is actually the cesium chloride density gradient that I talked about. You can see the position of this 15 NH4Cl incorporated into the DNA at the bottom. After 20 minutes, there was a formation of hybrid DNA. That is, one of the strand will have 14N DNA and the other strand will have 15N DNA. Okay. Now, again after 20 minutes, what happened? It led to the formation of two separate 14N DNA strands and two of these hybrid DNA strands. That is, one strand will contain 15N DNA and the other strand of the DNA helix will contain 14N DNA. And this was the position. You can see that this hybrid one is actually present in an intermediate position. This 14N is present above it and the 15N, as you can see, is situated below it. So what does it prove actually? From this original parental 15N DNA strand, it had two helices. Those helices actually separated and then this new helix which were formed, it was placed in 14NH4Cl medium. So this new strand contained this 14NH4Cl. But this old strand which came from the parental DNA strand, this contained 15NH4Cl. That is why the two strands were of different composition. So what can you say from that? One of the strands were actually coming from parent and one of the strand was actually the newer one which was being formed. Therefore, this is a semi-conservative method of DNA replication where one of the strand came from the parent and from using that parental strand as template, a newer strand was formed. So the DNA which was extracted from the culture 1, after 20 minutes, it had a hybrid or intermediate density. That is, one strand composed of 14N and the other strand composed of 15N. Now the DNA extracted from the culture after 40 minutes had equal amounts of hybrid and light DNA. That is 50% of hybrid character. Now there is a question, these questions are very important from NEET perspective. That is, if the E. coli was allowed to grow for 80 minutes, what would happen? So you can see that after first 20 minutes, the hybrid character was actually 100%. After 40 minutes, the hybrid character was reduced to 50%. After another 20 minutes, that is after 60 minutes, the hybrid character was reduced to 25%. And then in 80 minutes, the hybrid character will be reduced to 12.5%. So actually by every 20 minutes, the hybrid character is actually getting reduced to its original half. I hope you can get it. Now this actually proves that DNA replication is semi-conservative. Now very similar experiments were actually also done by Taylor and his colleagues. You have to remember the year 1958. Using radioactive thymidine to detect distribution of the newly synthesized DNA in the chromosomes on Visia faba, that is faba beans. They also proved that the DNA in chromosomes also replicate semi-conservatively.